The nerve supply and sensory receptors found in the skin. Now, efferent sensory nerves enter the skin, and these nerves end at epithelial and dermal receptors. The sweaty glands, the erector pili muscle, and the blood vessels are supplied by postganglionic sympathetic nerves. There are no parasympathetic nerves that reach the skin. Now, due to the large surface area of the skin and its external location, the skin is very suitable to function as a receiver of various stimuli from the environment, can detect various stimuli from the environment. To serve this function, various sensory receptors are found in the skin. Some have glial and/or collagenous covering, meaning that they are surrounded by a capsule made of glia cells or connective tissue, and some are unencapsulated. They do not have a capsule. If you look at this image here, for example, we have free nerve endings, we have tactile discs, and the root hair plexus. These are all unencapsulated nerves. We have Meissner or tactile corpuscle. We have the lamellated or Pacinian corpuscle. We have Ruffini corpuscle and Krauss bulbs. These are all capsulated receptors. These are all capsulated receptors. And we'll talk briefly about each of these types. So we start with the unencapsulated receptors. These are not covered by glial or collagenous capsule, and they include the tactile discs, which are associated with the epidermal tactile cells. Of course, these are Merkel cells, and as we have said, these are responsible for the sensation of light touch. Also, we have free nerve endings found in the papillodermis and even extending into the lower epidermal layers. These respond to temperature, pain, and itching. And we have the root hair plexus. This is a web of sensory fibers surrounding the bases of hair follicles. These are found in the reticulodermis. These detect movement of hairs, so that if the hair moves for any reason, these nerves will detect this movement. All these are unencapsulated. They are not surrounded by a capsule. The encapsulated receptors, there are several encapsulated receptors in the skin. We start with Meissner corpuscles. In this image here, this part is a dermal papilla, which means that this part here, the basophilic region, this is an epidermal ridge. Within the dermal papilla, we have these elliptical structures. And these are Meissner corpuscles. So Meissner corpuscles are elliptical structures located in the dermal papillae perpendicular to the epidermis. If this is the direction of the epidermis, here, then the Meissner corpuscle will be perpendicular to it. Notice, it's perpendicular to the direction of the epidermis. Meissner corpuscles are formed of a fibrous capsule on the outside. Here on the outside we have a capsule, and within it we have several flattened Schwann cells. These are arranged as lamellae. So here you have a Schwann cell, another Schwann cell. They are stacked. These Schwann cells are flattened and stacked like lamellae on each other, and so on. An unmyelinated nerve ending will wind among the Schwann cells. So a nerve ending will pass, becomes unmyelinated, it will wind between these flattened lamellae of Schwann cells until it reaches the tip of the receptor. Meissner's corpuscles are responsible for the sensation of light touch, and they are most numerous in the fingertips, palms, and soles. Notice how we said that Merkel cells, and here we said that Meissner corpuscles are most numerously found in the fingertips, and they are both responsible for the sensation of light touch. Remember that we use our fingertips to sense objects. When we carry something, when we touch an object, we sense the features of that object with our fingertips, and this is why we need a lot of receptors for light touch in this area. Laminated or Pacinian corpuscles. These are large oval structures located deep in the reticulodermis and the hypodermis. These receptors are formed of a fibrous capsule on the outside. You have a fibrous capsule, and in the middle we have an unmyelinated sensory fiber. Now, this sensory fiber is surrounded by several lamellae, and these lamellae are actually formed of two parts. This part here that immediately surrounds the unmyelinated fiber is actually formed of several concentric layers of flattened Schwann cells. The inner lamellae immediately surrounding the nerve. On the other hand, outside of this part, outside of this region, the outer lamellae are layers of connective tissue. So the lamellated corpuscle has a nerve fiber in the center. This nerve fiber is surrounded by several layers of flattened Schwann cells, and these flattened Schwann cells are in turn surrounded by several layers of connective tissue, and all the structure is surrounded by a fibrous capsule. The laminated or Pacinian corpuscles are responsible for the sensation of deep touch, pressure, and vibration, and these structures may also be found in other organs. Ruffini corpuscle are elongated fusiform structures found in the dermis. Notice here, this image, it is a fusiform structure. It is formed of a thin capsule. Here we have a thin capsule. Now this thin capsule is attached to the surrounding tissue. There are several attachments here attaching the capsule, collagen fibers attaching the capsule to the surrounding dermis. Within the capsule, we have a fluid-filled interior and an unmyelinated axon that branches profusely inside the capsule. Notice how on the inside the nerve fiber is unmyelinated and branches a lot. It responds to stretch. Whenever the dermis is stretched, this receptor will respond by generating a nerve impulse. Crowns and bulbs, these are ovoid structures here in this image, with extremely thin capsule. It is found in the skin of the penis and clitoris. It is found in several mucous membranes and even in the epineurium of nerves. It is found in the epineurium of nerves. It responds to vibration. The subcutaneous layer, or the hypodermis. We said that the skin is formed of two layers, the epidermis and dermis. Below the skin, we have a layer called the subcutaneous layer or the hypodermis. Here in this image, this is the subcutaneous layer. It is a loose connective tissue layer. It binds the skin to underlying tissue. It contains adipocytes and a vascular plexus. It is filled with adipocytes, so it's sometimes considered a fatty tissue. It is considered a fatty tissue. What are the functions of the subcutaneous layer? It enables the skin to slide over underlying structure. Because it is loose, it enables the skin to slide over underlying structures. The fat in this layer act as energy storage site, insulator, and shock absorber. So first of all, this layer plays a metabolic role by storing energy in the form of fat. And it is an insulator because it contains fat, and fat can act as an insulator. The rich vasculature, there is a vascular plexus here, the subcutaneous vascular plexus, This will enable the rapid intake of drugs injected into this layer. So sometimes we inject drugs directly into the subcutaneous layer, and in this way we can ensure the rapid delivery of this drug into the circulation. The hair. Hairs are elongated keratinized structures derived from epidermal invaginations. So this is one of the derivatives of the epidermis. Hairs are found throughout the body, except in certain areas like the palms, the soles, the lips, the glans penis, clitoris, and the labia minora. Hair distribution differs according to genetic factors, to gender, and region of the body. For example, over the head we have a lot of hair, the faces of males are hairy, and other regions have different hair distribution. Functions. Protection. For example, the hair on the head protects the scalp, the eyelashes and the eyebrows protect the eye. Also, reduction of heat loss and sensation of light touch. As we have said earlier, we have a hair root plexus of nerves surrounding the hair follicle that detects movement of hair. What are the parts of the hair? The hair is formed of the shaft, which is the superficial part of the hair that extends beyond the surface of the skin. So it is the part that is found outside the skin. The root is the deeper part of the hair that extends deep into the dermis, reaches down deep into the dermis, and it is surrounded by hair follicle. The hair root is surrounded by the hair follicle. The hair is actually formed of three concentric layers. First, we have the medulla in the middle, the innermost layer of large vacuolated cells. Then we have the cortex surrounding the medulla. It is the middle layer formed of cuboidal cells. And on the outside, we have the cuticle. The cuticle is formed of heavily keratinized squamous cells that are arranged like shingles on house roofs. Look at the cells here and how they are arranged, just like shingles. يشبهون ترتيب القرميد على أسطح المنزل. Now we said that the hair root is surrounded by the hair follicle. What is the hair follicle? The hair follicle is a downward extension of the epidermis that surrounds the hair root. So this is why we said that the hair is derived from the epidermis, because the hair follicle is derived from the epidermis. Now let's turn to the image. Let's look at this image here. Here in the middle we have the hair root, and all this is the hair follicle. It completely surrounds the hair root. If we take a cross section through this region, we'll get this image here. So what are the layers of the hair follicle? Because the hair follicle is formed of several layers. So first of all, let's look at the hair. In the middle here, we have the medulla surrounded by the cortex, surrounded by the cuticle. Now, this is the structures that are found in the hair, the parts of the hair. What about the hair follicle? The hair follicle is formed of an epithelial root sheath, and this is derived from the epidermis. This is derived from the epidermis. It is actually formed of two parts. The inner part, this part here, called the internal root sheath. This part, however, disappears above the attached sebaceous gland. A sebaceous gland usually attaches itself to the hair, and this internal sheath will disappear above this level. So it is present here, but above here the internal root sheath is not present. Then we have the external root sheath, This one extends to the epidermis, the entire length of the hair to the epidermis, where it becomes continuous with the stratum basale and the stratum spinosum. The external root sheath eventually reaches the epidermis and becomes continuous with the stratum basale and spinosum. After that, we have the glassy membrane. In this image, it appears as this purple line. The glassy membrane is actually the thickened basement membrane that separates the epithelial and dermal root sheaths. Remember when we said that under the epithelium we have a basement membrane. Remember, and the epithelial root sheath is derived from the epidermis, so it is formed of epithelial cells. So surrounding the epithelial root sheath we have a basement membrane. And here it is thickened, and it is called the glassy membrane. Finally, we have the dermal root sheath. This is a connective tissue sheath derived from the surrounding dermis. So, again, the hair follicle is formed of an epithelial root sheath, then a glassy membrane, and then a dermal root sheath. Now, if we look at the image here, we see that this part here is dilated. This part is dilated. This is called the hair bulb. Now, the hair bulb is invaginated. It is invaginated by what we call the hair dermal papilla. The hair dermal papilla is formed of loose areolar connective tissue containing capillary loop. And this capillary loop is important for the nourishment of the growing hair. Also, in the hair bulb, we have the hair matrix. This is the hair matrix. The hair matrix is formed of keratinocytes that are continuous with the stratum basale of the epidermis. Now remember, the stratum basale is the layer that forms all the other layers of the epidermis. Now the hair matrix, which is continuous with the stratum basale, is the source of the hair. It is the hair matrix is the part that will form the hair. And it contains melanocytes. The hair matrix contains melanocytes that are responsible for the color of the hair. And also remember, we said earlier that the hair bulb is surrounded by the hair root plexus, which detects movement of the hair. The erector pili muscle is a bundle of smooth muscle cells. These extend from the superficial part of the dermis to the dermal sheath of the hair follicle. So here, this is the dermal sheath, and this is the superficial part of the dermis, and here we have the erector pili muscle. Now, under stressful conditions, for example, in cold conditions, the muscle contracts. When it contracts, it will pull the hair so that the shaft will become perpendicular to the surface of the skin. And also, when this is done, the skin surrounding this region, surrounding the hair, will bulge, leading to the formation of what we call goosebumps. So notice how the shaft is perpendicular and the skin here is bulged, forming goosebumps. Now, in animals that have a lot of hair, this is beneficial to trap. When this happens, warm air will be trapped between the erected hairs, thus keeping the body warm. In humans, because they have less hair than animals, the exact reason for why this is happening is not clear, because in humans, because they have less hair, it is not very effective in warming the body, this reaction. So why does this reaction happens in humans is not exactly known. Hair growth is cyclical. It is asynchronous, meaning it doesn't happen at the same time, and it occurs at different rates in different regions of the body and even in the same region. So imagine the same region or different regions, different hairs grow at different rates and in different times. Now, growth of hair passes into 3 phases. Anagene is a generally long period of mitotic activity and growth. So the hair bulb is large, and the hair is growing. Catagene is a brief period of arrested growth and aggression of the hair bulb. Here the growth will be slowed down and become arrested, and even the hair follicle will become smaller and smaller progressively. Telogene, the final phase, is a long period of inactivity in which the hair may be shed. So here, the hair follicle has stopped working at all. It is not forming new hair at all. And because of that, there will be no further nourishment of the hair, and eventually it will fall off. Of course, even after that, a new phase will begin. A new anagene phase will start. Sebaceous glands are simple, branched, acinar, holocrine glands. They're found in the angle between the erector pili muscle and the hair follicle. Now, the sebaceous gland with the hair follicle form what we call the pilosebaceous unit. We have put these two together because actually the sebaceous gland and the hair follicle are derived from the same stem cells. Sebaceous gland are found in most regions of the body, with highest density in the face and scalp. However, they are absent in the palms and the soles. The duct of the gland open into the hair follicle. In certain regions, however, for example, the eyelids, the nipples, the penis and clitoris, the duct opens directly onto the epidermal surface. Now, secretion of these glands is called sebum, and it is mostly formed of lipids. And because this is called sebum, the cells that produce them are called sebocytes. Sebum helps maintain the stratum corneum and the hair shaft, and it has some antibacterial. If you look at this image here we have the hair, and this is the hair follicle. Notice how this is the sebaceous gland opening into the hair follicle, and look at the position of the sebaceous gland. This is the hair follicle, and this is the erector pili muscle, and the sebaceous gland is located at the angle between them. If you look at the histological section here, this is the sebaceous gland. It is surrounded by a capsule. And look at the cells. The cells have a white appearance. They appear empty. Remember, we said that sebum is mainly formed of lipids. This is why the cells appear empty. An important condition related to this gland is acne vulgaris. Acne vulgaris. Acne vulgaris is an inflammatory disorder of the pilosebaceous unit. So this is a disorder of the sebaceous gland and the hair follicle. In this condition, we have excessive keratinization and excessive sebum production. So the hair follicle, there will be excessive keratinization so that it becomes blocked, and we have excessive sebum secretion. So eventually the duct of the gland will be blocked. This will lead to accumulation of the sebum inside, as we can see here. The sebum has accumulated, and this accumulated sebum may be infected. This most commonly occurs during adolescence. ذلك يسمى حب الشباب. An interesting fact is the fact that a small microscopic mite called Demodex follicularum actually lives normally in the hair follicle of humans. Every adult human around the world, they have this microscopic mite living inside the hair follicle, Demodex follicularum. And this is it under the microscope. It feeds on dead cells and sebum. So far, it lives normally in humans. It is thought to be implicated in some conditions, in some skin disorders. However, nothing has been proven yet. There are certain skin diseases in which scientists think that this might be the cause, but nothing has been proven about that yet. Now talk about sweat glands. Sweat glands are long epidermal invaginations. So again, sweat glands are derived from the epidermis. They are simple coiled mucocrine glands of two types. Although there are two types of sweaty glands, both of these types are simple coiled mucocrine glands. Let's talk about each of these types. We start with the ecrine, the first type, ecrine sweaty glands. These are found all over the body, with highest density in the palms and soles. These play an important role in thermoregulation with some excretory function. So these help to preserve body heat, or they help to cool the body. So they play a role in thermoregulation. Also, the body can excrete certain substances with the sweat through these glands. The eccrine sweaty glands are controlled by the sympathetic nervous system, and they start to function from birth. Now, the secretory portion has a small lumen with stratified arrangement of cells. We'll see that in a few moments. The coil duct is lined by two layers of more eosinophilic epithelial cells. Why are they eosinophilic? Because they are filled with mitochondria. The cells that line the duct of these glands are highly eosinophilic because the cytoplasm of these cells is filled with mitochondria. Why do we need mitochondria in the duct? Because these ducts absorb sodium ions. The cells that line the ducts of the sweaty glands absorb sodium ions to prevent excessive loss of this electrolyte. So this is one of the functions of these sweaty glands, is to excrete excess sodium. But to prevent excessive loss of this electrolyte, some of the sodium is reabsorbed from the sweat by the cells of the ducts. In order to do that, they require pumps, and pumps require energy, and therefore there are a lot of mitochondria in these cells. The duct of these glands open into the surface of the skin at sweat pores. So there are three types of cells in the secretory portion of the eccrine sweaty glands. First type is called clear cells. Clear cells. These are, from the name, pale staining, and they have numerous mitochondria. Now, microvilli and basal enfoldings increase the surface area. So they have microvilli on their apical surface and basal enfoldings on the basal surface. These cells do not reach the lumen. They do not reach the lumen. So the cells are not in contact with the lumen. So how does the secretion of these cells reach the lumen? Now, interstitial fluid from the nearby dermis is taken up by these cells and released into intercellular canaliculi, small spaces between the cells, and these spaces eventually lead to the lumen. The clear cells of the eccrine sweaty glands secrete the water and electrolyte component of sweats. So this is important. We have to understand this. These cells take up fluid from the nearby tissue, and they take the water and electrolytes from this fluid and excrete it into intercellular canaliculi, and from there they are released to the outside, into the lumen. These are the clear cells. The dark cells, the second type of cells, these are inverted cone-shaped cells. They reach the lumen but do not rest on the basal lamina. Remember, we said these cells have stratified arrangement. So some of them rest on the basal lamina and some don't. Again, they reach the lumen, the dark cells, but do not rest on the basal lamina. The cytoplasm contains mitochondria, Golgi apparatus, and rough endoplasmic reticulum, which indicate that these cells produce proteins. Now, the wide apical part of the cell is filled with strongly eosinophilic granules. Why? Because these cells secrete or release glycoproteins by exocytosis into the lumen. So this is why we said they are protein-producing cells, because they produce glycoproteins, And this is why the granules are eosinophilic, because they contain proteins. Now, these cells release the glycoproteins into the lumen by exocytosis, and therefore these glands are merocrine. These glands, as we have said, are merocrine. The last type of cells found in the secretory portion of the eccrine sweaty glands are the myoepithelial cells. These cells rest on the basal lamina, and as you know, these cells contract to help push sweat into the ducts. If you look at this image here, we have the secretory portion of an eccrine sweaty gland, and here is the duct. Notice how both of them are coiled, the secretory portion and the duct, and they open on the surface of the skin at a sweat pore. If you look at this image here, these are the types of cells found in the secretory portion of the eccrine sweaty glands. The dark ones, the large dark ones, these are the dark cells. Notice how they have a wide apical part and a narrow basal part. Then we have the clear cells. These are the clear cells. Notice how these cells have basal enfoldings, and this is the intercellular space. This is the intercellular space, and here we have microvilli. And finally, these are the myoepithelial cells. Notice how they rest on this basal lamina. This is the basal lamina, and the myoepithelial cells rest on the basal lamina. So these are the three types of cells found in the eccrine sweaty gland secretory portion. The second type of sweaty glands is the apocrine sweaty glands. The apocrine sweat glands. These are found in the axillary and perianal regions of the body. They are controlled by the sympathetic nervous system and by sex hormones, and these glands become fully functional after puberty. Now, the secretory portion has a large lumen. We have a large lumen here with simple cuboidal eosinophilic cells. So here the cells are arranged in only one layer. The duct opens into the hair follicle near the epidermis. Now, these glands produce a viscous protein-rich secretion, and it is known that in animals, these glands produce a type of hormones called the pheromones. In humans, it is thought that they also produce pheromones, but in a reduced manner, to a lesser extent. Now, the secretion of these glands is odorless. It doesn't have an odor. However, bacteria will act on this secretion, giving it a distinctive odor, and this is what we call body odor. Each person has a distinctive odor to his body. This odor, this smell, is caused by the action of bacteria on the secretion of the apocrine sweaty glands. Now, note, when first discovered, these glands were thought to be apocrine, meaning that they produce their secretion in an apocrine method, and they were named so, apocrine sweaty glands. However, throughout the years, research have shown that these glands actually release their secretions by exocytosis, and therefore are merocrine glands rather than apocrine. So although this is called an apocrine sweaty glands, they are merocrine glands. They are merocrine glands. If you look at the image here, this is the secretory portion of an apocrine sweaty gland, and here we have the duct, which opens into the hair follicle. Opens into the hair follicle close to the epidermis. Here in this image, this is the secretory portion of an eccrine sweaty gland. Notice the small lumen. compare with the secretory portion of an apocrine sweat gland. Here we have a large lumen. Also, the epithelium is thinner because in the apocrine it is simple, whereas in the acrine it is stratified. Also here, on this side, this is a duct. Notice the dark red color. As we have said, the epithelial cells of the duct are highly eosinophilic. Now, this table shows a quick comparison between the acrine and apocrine sweaty glands. We have talked about these, all of these points. What's important is that both of them are merocrine. We have to remember both of them are merocrine. And also, acrine sweaty glands are controlled by cholinergic nerves, whereas apocrine are controlled by adrenergic nerve, in addition to hormones. And therefore, apocrine sweaty glands are mostly stimulated during emotional stress. The last topic is nails. Nails are hard plates of keratinized cells found on the dorsal surface of the distal phalanges. Externally, as can be seen in this image, the nail is formed of a nail plate. This is the plate. This plate is surrounded by 3 folds of skin. This is one lateral fold, and another lateral fold, and a proximal fold. So I have 3 folds of skin. In addition, the nail has a free edge. This is the free edge of the nail, the part that we clip, the part that we cut. This is called the free edge. Now the proximal part of the nail, which is called the nail root, this is the part that is found under, that is covered by the proximal fold. So this is the proximal fold, so it is found under this region. At this fold, the stratum corneum does not pass with the other layers of the epidermis, but continues distally as the eponychium, also called the cuticle. So here the eponychium, this whitish, very thin white line found proximally, this is actually the stratum corneum. This is actually stratum corneum. Now the nail root is formed from the nail matrix, and the nail matrix is derived from the epidermis. So again, the nail is a derivative of the epidermis, another derivative of the epidermis. As more cells are added, the root grows and the plate extends distally over what we call the nail bed. So the part over which the nail grows is called the nail bed. In the proximal part of the nail plate, there is a white crescent-shaped area called the lunula. If you look at, for example, the fingernail of your thumb, which is very obvious, you'll see in the proximal part a crescent-shaped white area. This is called the lunula. The rest of the nail is pinkish. Why is that? The white color of the lunula comes from the opaque nail matrix under the lunula. The matrix that forms the nail plate is thick, and therefore the nail plate above it is white in color. The nail bed, on the other hand, is very thin, formed of the stratum basale and spinosum only. So the nail bed is very thin, formed of only these 2 layers. Because of this, and because the mature nail plate is semi-transparent, the color of the blood in the dermal vessels can be seen through the nail. And this is why the nail plate appears pinkish in color, because we can see the blood through it. Now, if we have a state of deoxygenation, we can see that in severe cases of deoxygenation, we can see that through the nail. We can see bluish color through the nail. Distally, the free edge of the nail is adhered to the fingertip by a fold of epidermis called the hyponychium. Hyponychium. Let's look at this image. This is the nail plate, and here we have the proximal fold, the proximal skin fold. It is formed of all the layers of the epidermis. This fold, the epidermis here, from its name, it will fold. The layers of the epidermis will fold. However, the stratum corneum will not fold, but it will continue distally here to form the eponychium, The other layers of the epidermis will continue to fold to form what we call the nail matrix. Okay? So this is here the nail matrix. This is the part that forms the nail. The initial part of the nail, this is the nail root. The first part that is formed by the nail matrix here in this region, this is the nail root. More cells are added in this direction. And the nail will grow in this direction, will grow distally. The nail will grow distally. The area over which the nail grows, this part here, this is the nail bed. Initially, we have in the nail matrix, this part, as you can see, this is thick. This part here is thick. And therefore, this region of the nail plate will appear white in color. And this is the lunula. The nail bed, on the other hand, is thin. As you can see, this part here, it is thin, formed of only the stratum basale and spinosum. And therefore, the blood vessels found here in the dermis here, the blood in them can be seen through the nail, Here, the finger has ended, and this part of the nail is the free edge, but it is attached to the fingertip by the hyponychium. Finally, fingernails grow at a faster rate than toenails, and this is why we clip our fingernails more often than toenails, because they grow faster. And by this we end the topic of histology of the skin, and I hope you have all the success in the world, and I hope you enjoyed these lectures. Thank you.